The role of gut microbiota in immune system regulation. Most people think about the gut mainly as part of digestion. Food goes in, nutrients are absorbed, waste leaves the body, but the gut is connected to much more. Inside the intestines live trillions of microorganisms, including bacteria, fungi, and viruses, called the gut microbiome. These microorganisms are not passive. They interact constantly with the immune system, influencing how the body responds to threats, inflammation, and daily regulation. Gut microbiota refers to the community of microorganisms living mainly in the large intestine. Different species perform different functions. Some break down foods, some produce useful compounds, and others interact with immune cells. The body and these microorganisms exist in a partnership. Humans provide environment and nutrients, while microbiota regulate processes that support health. This relationship is more important than realized. The immune system has a difficult job. It must defend against harmful pathogens while avoiding attacks on harmless substances or own tissues. Balance matters. An overactive response causes chronic inflammation or autoimmune disease. An underactive response increases vulnerability to infection. Gut microbiota help regulate this balance. How does the immune system learn the difference between danger and normal biological activity? Part of the answer involves constant interaction with microbes in the gut. The lining of the intestines contains large amounts of immune tissue. Immune cells continuously monitor what passes through the gut. Certain gut bacteria produce signals and compounds that influence immune cells. Some reduce inflammation, others stimulate protective immune responses. This communication happens constantly, even when people are not aware of it. One major idea in microbiome research is a diverse microbiota means many different species are present, not just a few. Diversity matters because different microbes perform different roles. Some maintain the intestinal barrier, some compete against harmful bacteria, and others influence immune regulation. When diversity decreases significantly, the system may become less stable. That does not mean every person needs a perfect microbiome, but it does show how balance and variety affects biological function. Think about what often happens after taking strong antibiotics. Antibiotics can kill harmful bacteria, but they may also reduce helpful bacteria temporarily. Sometimes digestion changes afterward, or people feel physically different. That experience shows something important. The microorganisms inside the gut are not biologically irrelevant. Changing them changes how the body functions too. The gut lining acts like a barrier separating the inside of the body from substances moving through the digestive tract. Certain gut bacteria help maintain this barrier by supporting the cells lining the intestines. If the barrier becomes disrupted, unwanted substances may interact with the immune system. This is interesting because the immune system is not only reacting to pathogens, it is also responding to how effectively the gut environment is being maintained. Why inflammation connects to the microbiome? Inflammation is a normal part of immune defense, but chronic inflammation can contribute to disease over time. Gut microbiota influence inflammatory signaling through multiple pathways. Some bacteria produce short-chain fatty acids, which can help regulate immune activity. Others may contribute to inflammatory responses under certain conditions. The balance between these microbial populations partly affects how the immune system behaves. The relationship between diet and gut microbiota diet strongly affects gut microbiota composition. Different microbes thrive on different nutrients. Diets rich in diverse plant fibers often support bacteria that produce beneficial compounds during fermentation. Highly processed diets may influence microbial balance differently. This does not mean one meal suddenly transforms the microbiome overnight, but repeated dietary patterns shape the environment over time. The body is constantly responding to what repeatedly enters the gut. Why early life matters so much? The microbiome begins developing early in life. It changes over time through diet, environment, illness, and other exposures. Early microbial exposure may influence immune system development significantly. This is one reason researchers study how microbiota affect allergy risk, autoimmune disease, and immune regulation later in life. The immune system is not developing in isolation. It develops while interacting continuously with microbes from the environment and the body itself. The gut-brain immune connection, another area receiving attention, is the gut-brain immune connection. Gut microbiota may influence signaling involving the nervous system, hormones, and immune activity. This area is still developing scientifically, but it suggests the gut may affect more than digestion and immunity alone. That part feels slightly strange at first because people often separate body systems mentally. But biologically, the systems communicate constantly. Why scientists are careful about oversimplification microbiome research became extremely popular online, and sometimes the science gets oversimplified. People may hear phrases like gut health controls everything, which is probably too broad. The microbiome is important, but it is also highly complex. Different people can have different microbial compositions while still being healthy. Researchers are still learning exactly how specific microbes influence disease, immunity, and regulation. That uncertainty matters because the science is still evolving. The difference between correlation and cause. One challenge in microbiome research is distinguishing correlation from direct cause. Certain diseases are associated with changes in gut microbiota, but that does not always mean the microbiota caused the disease originally. Sometimes illness itself changes the microbiome too. This is important because biological systems influence each other continuously. Researchers must carefully determine whether microbial changes are drivers of disease, responses to disease, or part of both processes together. Why the future of microbiome research matters, scientists are exploring whether microbiome-based therapies could eventually help regulate immune-related conditions. Probiotics, prebiotics, dietary interventions, and targeted microbial therapies are all being studied. But the future will require much more precision. The microbiome is an ecosystem, not one single organism. Understanding that complexity is part of what makes this field both promising and difficult. Final thoughts: gut microbiota play an important role in immune system regulation by interacting constantly with immune cells, supporting intestinal barriers, influencing inflammation, and helping maintain biological balance. The gut is not only a digestive organ, it is part of a larger communication system, connecting microbes, immunity, metabolism, and overall physiology. Once you understand how much activity is happening inside the gut continuously, the relationship between microbiota and immune regulation starts feeling far more important than most people originally assume.